Now let's begin our review of neuroscience and emphasizing the clinical and anatomical functional aspects of the central nervous system that are commonly seen. First topic we want to deal with before we get into the clinical anatomy is how the central and peripheral nervous systems develop, a process known as neurulation. And the key points involving this are illustrated on this first image. First point is neurulation begins just after gastrulation in the third week. And this process or timeline is important because gastrulation established the three primary germ layers, ectoderm, mesoderm, and endoderm, because a structure in the mesoderm, the notochord, has to be present to induce the overlying ectoderm to begin the neurulation process. So that's the point that's made here. Neurulation begins in the third week. The notochord, which develops in the mesoderm, induces the overlying ectoderm to form the neural plate. And the neural plate is neuroectoderm that forms in the future dorsal midline of the embryo. That neural plate then invaginates to form a neural groove that is separated by a pair of neural folds. By the end of the third week, the key event in neurulation has taken place where the lateral lips of those neural folds grow toward the midline, fuse in the future dorsal midline, and form the neural tube. And in conjunction with this, as we'll see, neural crest cells also form from neuroectoderm, but they form outside the neural tube at the lateral lips of the neural plate. So now an illustration that documents this process is on the next image. First off, on the top right of this image, you can see a cross-section through the developing embryo that illustrates the three primary germ layers, ectoderm, mesoderm, and endoderm, obviously existing just post-gastrulation. Also, you can see that in the future dorsal midline, in the mesoderm, is the notochord, a structure which will persist in postnatal life as the nucleus pulposus in the middle of an intervertebral disc. So now, tie this in conjunction with the image that's on the upper left that shows a cross-section at A, which illustrates, again, our three primary germ layers and the site where neurulation takes place. So the first stage of neurulation is an induction essentially performed by the notochord, which induces the overlying ectoderm in the future dorsal midline of the embryo to form neural ectoderm. That neural ectoderm forms initially a neural plate. Then it invaginates in the midline to form a neural groove separated by a pair of neural folds. And then if you look down at the image at letter C, you can see now that the two lateral lips of the neural groove have fused together in the dorsal midline to form a completely hollow neural tube. Note also in the image at C that the neural crest cells are developing outside the neural tube and will remain in that location. So now, over on the lower left of this image is the schema that illustrates the time course of neural tube closure. And the point to be made here first is that the middle section of the neural tube at C closes first. Then in proper 2-day sequence, the rostral neural pore closes second, and then finally the caudal neural pore closes third and last. So now, basically, when there are defects in neurulation, they are commonly defects in the closure of either the rostral end of the neural tube or the caudal end of the neural tube. And there are different terminologies that describe this. First off, if the rostral neuropore fails to close, the condition that results is known as anencephaly. When the caudal neuropore fails to close, it gives rise to the most severe form of spina bifida. So now, in either of these neural tube defects, there will be elevated levels of alpha-fetoprotein detectable in during amniocentesis. But the distinguishing factor is, other than utilizing ultrasound imaging to visualize the defect, note that fetuses that have a rostral neuropore defect, polyhydramnios will also be complicating the pregnancy. But in contrast, when a fetus has a caudal neuropore defect, a form of spina bifida, there will be no or normal levels of amniotic fluid. So recognize that that is a distinguishing feature between the two. So now the reason being is that the rostral neuropore provides the neural mechanisms that control swallowing, which is why polyhydramnios will be evident during pregnancy. The caudal neuropore, which will form the caudal end of the spinal cord, basically has nothing to do with the swallowing mechanism. Therefore, normal levels of amniotic fluid will be present during the pregnancy. So now, in D is an illustration that points out that even at the earliest stage of neural tube closure, the neural tube has a polarity that's important to remember. Basically, as indicated by that horizontal red line, the neural tube fundamentally can be divided horizontally into a dorsally positioned alar plate and a ventrally located basal plate. The alar plate is basically going to form or give rise to neurons that respond to sensation, sensory neurons. The basal plate is going to contain motor neurons. And this fundamental plan will be replicated or persist in a way in the postnatal form of the spinal cord as well as the brainstem. So now, in summary, we can say overall that the neural tube is certainly going to form the central nervous system, including the spinal cord, and certainly the elements that are components of that will be what we'll see as or describe as lower motor neurons and preganglionic autonomic neurons, whose axons will form part of the peripheral nervous system. And as we alluded to already, the brainstem and spinal cord have an alar or sensory plate that's dorsally positioned and a basal plate that is ventrally positioned, and these two plates are separated by the embryonic sulcus limitans. So now, after the neural tube is formed, basically it undergoes a process of cephalization, where it enlarges dramatically to form initially 3 primary brain vesicles, and then subsequently 5 secondary brain vesicles. It is from the 5 secondary brain vesicles that all parts of the central nervous system are formed, except for the spinal cord. So let's summarize that, as well as including what elements will give rise to the peripheral nervous system. The peripheral nervous system basically is formed from 2 components. The neural crest cells, which we saw develop outside the neural tube, give rise to primary sensory neurons and all postganglionic autonomic neurons, and obviously their processes as well. They obviously will be found in various cranial and spinal nerves. So now, the peripheral nervous system per se certainly contains cranial nerves, 12 pairs, and 31 pairs of spinal nerves. Each of these nerves may be a combination of neurons derived from either neural crest cells or from axons of neural tube cells. So now, let's summarize overall what are the important elements to remember about embryonic origins of neural structures. The fundamental point on this image is to recognize that all of the neural system elements are derived from neuroectoderm that was induced to form by the underlying notochord. So now, basically, the neural tube, as we've said, forms basically all elements of the central nervous system, with some important additions that are listed here. Note that the optic nerve, the entire optic nerve, all the way out to and including the cells of the retina, is a direct outgrowth of the neural tube. And therefore, it's not truly a nerve because all of the myelin of its axon, since it's entirely contained inside the CNS, are formed by oligodendrocytes, which make all central nervous system myelin. As a result, and we point this out here, the optic nerve is the only cranial or spinal nerve that's going to be affected by a central nervous system demyelinating disorder, and most common of which being multiple sclerosis, that's abbreviated MS on this image. Note also that the central nervous system includes the pineal gland, as well as the posterior pituitary, and certainly included in the neural tube are a variety of glial cells. We've mentioned oligodendrocytes as those that make central nervous system myelin, but certainly the most prevalent glial cell in the CNS are the astrocytes, which, as we indicate here, control the microenvironment of neurons, help maintain the blood-brain barrier with their foot-like processes that are directly opposed to the capillary endothelial cells. And then finally, microglial cells are intrinsic to the central nervous system, but they migrate in, act as phagocytes. Since they are not part of the development of the central nervous system, they are part of the mononuclear phagocyte system. So now, one point to remember. Note that the pituitary gland has what we refer to as a dual embryonic origin. It's all derived from endoderm, but different kinds of endoderm. The neural hypophysis, or the posterior pituitary, has its neuron cell body situated inside the neural tube, such that only axons of those neurons that develop inside the neural tube are found in the neural hypophysis or posterior pituitary. In contrast, under a note on the right side of the page, the anterior pituitary is also derived from ectoderm, but is derived from oral ectoderm. And basically these cells form what is known as a Rathke's pouch diverticulum that grows dorsally into the sella turcica and is situated in close proximity to the posterior pituitary. But note that the anterior pituitary contains a multitude of cells that are giving rise and producing certain endocrine hormones of the anterior pituitary, but the posterior pituitary doesn't have any neurons in it because those neurons are found inside the neural tube. Now finally, let's make the very important distinction about what structures are derived from neural crest cells versus neural tube cells. First off, we can make a general statement that since neural crest cells form outside the neural tube, any neurons that are derived from neural crests are going to be found also outside the neural tube in ganglia. So we can make the generalization that all ganglia, and certainly all of them that are found in the peripheral nervous system, are derived from neural crest cells. And fundamentally below that, we indicate that there are only two types of ganglia in the body. One type of ganglion are sensory. They contain primary sensory neurons whose job it is to innervate a receptor of a wide variety of types and parlay that receptor information into the central nervous system. The other kind of neuron derived from neural crest cells are motor neurons, specifically postganglionic autonomic neurons, that obviously are going to be linked together synaptically with an axon of a preganglionic autonomic neuron whose cell body is found in the neural tube. So now, a specialized distribution of postganglionic sympathetic neurons are those that develop in the adrenal medulla. So the adrenal medulla, in essence, is a modified, specialized population of neural crest cells. Note, however, that the adrenal cortex is not derived from neural crests, it's derived from mesoderm. Additionally, neural crest cells form a wide variety of non-neuronal cell types that basically not only are not neurons, but they migrate out to far-reaching positions in the body, including not the least of which would be melanocytes that migrate out to the basal layers of the epidermis and produce myelin. Schwann cells are neural crest cell derivatives. As we know, the claim to fame of Schwann cells is to produce all myelin for axons in the peripheral nervous system. Two layers of the meninges are also derived from neural crests, pia and arachnoid. Note that the dura is derived from mesoderm. Pharyngeal arch cartilage is derived from neural crest, and pharyngeal arch cartilages are found in some of the pharyngeal arches that help contribute to the development of structures in the face and the visceral part of the neck. Note that virtually all other cartilage is derived from mesoderm as well. Odontoblasts form dentin in the teeth. Parafollicular cells are endocrine-producing cells that migrate into the thyroid gland and secrete calcitonin. And as you can see down below, the last two terms indicate that major components of the developing heart, the aorticopulmonary septum and the endocardial cushions, also develop from migrating neural crest cells. So now, let's take a further look at the developing neural tube and see its development into its component vesicles and what kind of postnatal central nervous system structures those vesicles form. On the left side of this image is the neural tube that has elaborated itself into three primary vesicles, the forebrain, the midbrain, and hindbrain. And you can certainly see below them the spinal cord extending inferiorly below the hindbrain. The 5 secondary brain vesicles are listed on the right side of this image, and you can see that of those 5, 2 of them are derived from the forebrain, the telencephalon and the diencephalon. 1 of them is derived from the midbrain, simply forming the mesencephalon, and the hindbrain also forms 2 secondary brain vesicles known as the metencephalon and the myelencephalon. And then, as we've already indicated, the spinal cord extends inferiorly from the myelencephalon. So now, before we talk about these structures in a little more detail, let's look specifically at what parts of the postnatal central nervous system are derived from the secondary brain vesicles. And this is illustrated on the chart at the bottom of the figure. So the telencephalon certainly gives rise to the largest components of the central nervous system, and they are the pair of cerebral hemispheres. But note also, as we'll see, that at the base of each hemisphere are major components of the basal ganglia. Diencephalon. Diencephalon gives rise to four structures that all end in the word thalamus. The thalamus literally means inner chamber, and there are four components to it. The major component, largest component of the diencephalon, forms the thalamus. And as we'll see, the thalamus contains nuclei that provide a filtering mechanism for any kind of input that is destined to reach a cerebral hemisphere. So putting it another way, we can say that the thalamus contains a collection of motor and sensory relay nuclei, whose job, as we said, is to filter all input neurally destined to reach a cerebral hemisphere. The other component, the other major component of the diencephalon is the hypothalamus. You can also see it on the list here. The hypothalamus basically is situated just below the thalamus and contains an array of mainly endocrine-producing cells. Closely tied and a direct outgrowth of the hypothalamus is the posterior pituitary. So the neurons that are producing posterior pituitary hormones are basically going to be found in the hypothalamus. Axons of those cell bodies are going to leave the hypothalamus and end up releasing their secretory products in the posterior pituitary. Finally, note that the retina, optic nerve, and all components therewith also are direct outgrowths of the brain, as we indicated earlier. But the point to be made here is the retina and the optic nerve are direct outgrowths of the diencephalon, hence the term, the eye on there. The last three secondary brain vesicles form the components of the brainstem, and they include the midbrain, the pons and the cerebellum, and the medulla. The midbrain certainly is derived from the mesencephalon. The pons and the cerebellum derive from the metencephalon, the medulla from the myelencephalon, and then, as we've indicated, continuing inferiorly below the medulla will be the spinal cord. The last point to be made on this chart is over on the right side. Note that since the neural tube formed as a hollow structure when the neural folds met in the midline, the neural tube maintains its hollow caliber throughout life. And what forms from that neural canal are elements of the ventricular system containing cerebral spinal fluid and the channels that interconnect them. So let's make the point here that in the telencephalon, in each one of the cerebral hemispheres will be a lateral ventricle. They communicate with a single third ventricle, which is found in the diencephalon. The cerebral aqueduct is found exclusively in the midbrain, meaning that the midbrain is the only part of or only component of the central nervous system derived from a secondary brain vesicle that does not contain a ventricle. The fourth ventricle is found overlying the pons between it and the cerebellum and extends down partially into the medulla. And finally, not illustrated on here is the fact that the spinal cord also contains a neural canal remnant, that being the so-called central canal. So now, let's also expand our discussion of neural tube defects by describing the different forms of spina bifida beginning on the next figure. So now, fundamentally, there are three different forms of spina bifida that are not true neural or open neural tube defects, because in A, B, and C on here, basically the neural tube, the caudal neuropore neural tube forms normally, and basically the defects are related to the fact that in addition to forming the caudal neuropore, the neural tube's job at this level is to induce sclerotome that forms components of the vertebral arch to grow around the developing spinal cord and form a definitive neural arch. So in reality, the first three forms of spina bifida illustrated on this figure are those that result from a defect in the overlying neural arches, resulting in missing spinous processes, and the presence of cyst-like protrusions through the defect. Finally, in D, we'll see a true caudal neuropore, open neuropore defect where the caudal neuropore fails to form. So let's describe these now individually, beginning with the least symptomatic or asymptomatic form of spina bifida, and this is known as spina bifida occulta. So now basically here, the only defect that is present is one or more missing spinous processes when the laminae of individual lumbosacral vertebrae fail to fuse in the midline. So now, in this condition, the spinal cord develops normally. It's in its normal position, situated within the components of the meninges. So basically, in this, as we point out here, there are no neurologic defects. And during the pregnancy, there will be normal levels of alpha-fetoprotein. So it's basically the asymptomatic, hence the term spina bifida occulta, the hidden form of spina bifida because of its lack of neurologic symptoms. So now, the next two forms of spina bifida are known as spina bifida cysticas. And basically, these are defined by the contents of a cyst-like protrusion that appears and is grossly visible in the midline of the lower back as a cyst-like protrusion between the defect and the neural arches. First off, a spina bifida with meningeocele presents with a cyst-like protrusion that is lined by the dura and arachnoid and contains cerebrospinal fluid. But since this is a dorsal midline body wall defect, elevated levels of alpha-fetoprotein will be detectable during amniocentesis. But this is a condition where, as you see in image B here, the spinal cord develops normally and is in its normal position adjacent to the bodies of lumbosacral vertebrae. So this condition is not going to result in any neurologic symptoms. In C is the more severe form of spina bifida cystica. This is known as spina bifida with meningeomyelocele. In this case, again, the dura and arachnoid protrude through the defect in the neural arches, and CSF is going to be the fluid component of the cyst. But in this case, the spinal cord is also displaced into the cyst, and this may result in a stretching of lumbosacral spinal nerves and maybe some altered sensation or motor weakness in the area of bladder function and the lower limbs. But again, since this is a true dorsal midline body wall defect caused by the cyst, there will be elevated levels of alpha-fetoprotein detectable during amniocentesis. Finally, in D, the true open caudal neuropore defect is known as spina bifida with myeloschisis. Here, basically, the neural tube literally fails to close in the caudal neuropore. It's the most severe variation, and basically there will be an open neural tube that lies on the surface of the developing back. This condition is probably not compatible with life because it's commonly seen with other congenital defects, and certainly since this is a well-documented body wall defect, there are going to be elevated levels of alpha-fetoprotein present. So the last point about spina bifida is if there are elevated levels of alpha-fetoprotein, it may indicate the presence of a neural tube defect and a form of spina bifida, but there may or may not be neurologic symptoms evident because in the less severe forms of spina bifida, elevated levels of AFP might be detectable, but there may not be signs and symptoms associated with neurologic defects. So now, let's next look at the structural components of a neuron. So now, many of you are certainly aware of basically the elements of a neuron. It's a typical eukaryotic cell in that it has a typical array of organelles, membrane and non-membrane bound. But a neuron is unique in the sense that it contains an array of long membrane-bound cytoplasmic processes. The most prominent of which are listed here are the dendrites, highly branched, whose job it is to basically receive afferent information destined to influence that particular neuron and transmit it to the cell body. The cell body is basically where we're going to find most of the subcellular organelles that are characteristic of a neuron. Probably the most clinically significant component of a neuron in terms of neurological disease is the axon. The single axon certainly's job is to transmit impulses down the length of it away from the cell body to the nerve terminals. The nerve terminals in a typical neuron contain synaptic vesicles, which contain neurotransmitter that is basically released into the synaptic cleft upon appropriate stimulation. So now if we look at a diagram of the neuron on this next image, we can certainly see the typical form of it, noting again the wide array of highly branched dendrites, the soma or cell body containing the typical range of subcellular organelles. So now, since this is largely a protein-producing cell, many of the proteins are going to be used as neurotransmitters. A typical neuron is going to have prominent amounts of rough endoplasmic reticulum, as indicated by the arrow. Rough endoplasmic reticulum, whose job is to synthesize either neurotransmitter or precursors of neurotransmitter, in a neuron is known as Nissl substance, as we indicate here. So now, the interesting feature about Nissl substance is that it is certainly found throughout the cell body and is also found in the proximal parts of dendrites, but Nissl substance is not found in any part of an axon. So the point is this. Axons utilize a phenomenon of axonal transport to transport newly synthesized neurotransmitter or its precursors down the length of the axon so it can be incorporated into synaptic vesicles, as illustrated at the nerve terminal, and released upon appropriate impulse arrival. So the directionality of axonal transport that is utilized to transport substances from the cell body to the axon terminal is known as anterograde axonal transport, and you can see it illustrated in the direction of the arrow. Axons also may utilize a phenomenon known as retrograde axonal transport, whose job it is to recycle membrane and maybe neurotransmitter synaptic vesicles that are picked up at the level of the of the nerve terminal. Retrograde axonal transport then transport substances to be recycled back to be degraded or recycled at the level of the cell body. So now, important point also about this shows a neuron whose cell body on the left side of the page is found in the central nervous system. And the axon of this neuron is leaving the central nervous system and is found on the right side of this image in the peripheral nervous system, certainly coursing in either a branch of a spinal or a cranial nerve. But this image gives us a good opportunity, again, to remind you that that axon, depending on where it is found, is going to be myelinated by a different form of glial cell. Central nervous system myelin is certainly going to be formed by oligodendrocytes. So the part of the axon that is emanating from the cell body until the point where it leaves the central nervous system, the boundary indicated by the dashed lines, all of that myelin of that axon will be formed by oligodendrocytes. In contrast, once the axon enters and courses through the peripheral nervous system in a nerve, the axon is going to be myelinated by Schwann cells, as indicated in this picture. So now, important distinction to make about the myelination is to predict if an axon is severed in either the central or the peripheral nervous system, what part of that axon may have the capacity to regenerate. And the answer is, if an axon is cut where its myelin is formed by oligodendrocytes, that axon in the central nervous system will not have the capacity to regenerate. In contrast, as we indicate here, if a cut axon is myelinated by Schwann cells, meaning it has been severed in the peripheral nervous system, that axon will have the capacity to regenerate by having the axonal sprouts regenerate down a tube of endoneurium, which basically is the sheath that is derived from Schwann cells that basically encircles and encases all of the either myelinated or unmyelinated peripheral nerve axons. So recognize that the endoneurial sheath of the Schwann cell is the most important element that permits the regeneration of axons, basically that in the peripheral nervous system that have been severed. So now finally, additional terminology. When you cut an axon, regardless of whether you sever it in the central or the peripheral nervous system, the part of the axon distal to the cut is going to degenerate because it has been cut off from its source of nutritional and trophic supply from the neuron cell body. So now, terminology. The directionality of that cut axon distal to the cut is known as anterograde degeneration, and a synonym for that is known as Wallerian degeneration. And whether, obviously, that axon will regenerate following its anterograde degeneration is simply a matter of whether its myelin was formed by Schwann cells, where it does have the capacity to regenerate, or oligodendrocytes, where it does not. So now, let's summarize the transport process, what subcellular organelles are facilitating it, and how it's mediated. Anterograde transport, as well as retrograde axonal transport, both utilize microtubules. But there are different ATPase motor molecules that drive the transport process. So you can see on this figure, anterograde axonal transport is driven by the ATPase motor molecule kinesin. And basically, as we indicated, helps move vesicles, neurotransmitter, proteins, so on and so forth, down the length of the axon from the cell body to the axon terminal. Retrograde axonal transport certainly also uses microtubules, but it's mediated by a different ATPase motor molecule, that of dynein. As we indicate, the retrograde axonal transport process is involved in recycling and transport substances to be degraded back to the lysosomal system. So now, unfortunately, exogenous substances such as certain viruses, herpes, polio, rabies, and tetanus toxin basically can affect neuron cell bodies as a direct result of them being picked up at the level of the axon terminal and transported back to the cell body unknowingly by the neuron utilizing retrograde axonal transport. So now, this is just a summary of the non-neuronal cells of the central nervous system, and we've summarized these previously, but let's make the distinction. Schwann cells certainly form myelin in the peripheral nervous system. The point is that a single Schwann cell simply innervates one individual section, an internode, of a single axon. So just remember that Schwann cells basically innervate a single segment of a single axon. Ependymal cells are going to be neurons or non-neuronal glial cells that line the ventricles and the central canal of the spinal cord. Microglia, as we've already indicated, are not derived from the neural tube, but migrate into it and basically act as phagocytes. Oligodendrocytes and astrocytes were summarized previously. Again, remember that oligodendrocytes can provide central nervous system myelin for a number of axons that are immediately adjacent to it. Next topic we want to discuss is the formation, flow, and resorption of cerebral spinal fluid and the clinical implications of disrupting that flow. First off, in this kind of phantom diagram, we again see an illustration of the location of the ventricles that represent remnants of the neural canal and their location. So this is instructive because you can see the form of all of these ventricles, beginning with the lateral ventricles. The lateral ventricles, as you can see, have basically a C-shaped configuration such that there is a component of the lateral ventricle in each of the lobes of a hemisphere. For example, the anterior horn of the lateral ventricle, as you can see here, projects up into the frontal lobe of each hemisphere. The body of the lateral ventricle is largely found in the parietal lobe. The posterior horn of the lateral ventricle, as we indicate, is found in the occipital lobe. And the inferior horn of the lateral ventricle basically projects down into each temporal lobe. So now, any CSF that is formed in a ventricle is produced by choroid plexus. As we indicated at the bottom of the slide, choroid plexus is found in all of the ventricles. It's found in the body and the inferior horn of each lateral ventricle. It's found in the third ventricle, and it's found in the fourth ventricle. So now, note how each third ventricle is linked to a lateral ventricle by an interventricular foramen of Monro, and you can see that short little opening joining those two areas on this image. The fourth ventricle is connected to the third ventricle by way of a cerebral aqueduct, which we identified as exclusively found in the midbrain. And then finally, extending inferiorly below the fourth ventricle, which is found between the pons and the overlying cerebellum, is the central canal that extends down into the spinal cord. So again, note that choroid plexus obviously is found in all of the ventricles and is secreting cerebrospinal fluid, which is in essence an ultrafiltrated blood plasma. So now the next point is just a summary that, again, that plexus is found in all of the ventricles and its job is to secrete cerebrospinal fluid. Now, in terms of CSF flow, we need to make a point, and the point is made in the middle of this slide. Cerebrospinal fluid can only leave the fourth ventricle to find its way into the subarachnoid space. So since that is the case, any CSF that is produced in either the lateral ventricles or the third ventricle must find its way down to the fourth ventricle to exit from the corresponding openings. So the flow of CSF is illustrated in text at the top of the image and then diagrammatically on the right. And the point is this: any CSF that's produced in either of the lateral ventricles must pass through an interventricular foramen of Monro into the single third ventricle. Any CSF that's collecting in the third ventricle must course down through the cerebral aqueduct to the fourth ventricle. And the fourth ventricle contains 3 openings that allow CSF to exit and dump into the subarachnoid space. They are the single midline foramen of Magendie, which is illustrated on this slide, as well as a pair of foramina of Luschka that exit laterally from corresponding points in the fourth ventricle. And then finally, once CSF enters the subarachnoid space, it circulates completely around all aspects of the central nervous system, obviously extending below the inferior extent of the spinal cord where lumbar punctures can be performed. The sites where CSF is absorbed back into the venous system occur at the level of arachnoid granulations, many of them protruding into the level of the superior sagittal durovenous sinus and returning CSF to that point. And if we look at a diagram, a coronal section, you can see in this section the location of the arachnoid granulations as being protrusions of the subarachnoid space, where basically CSF is passing down its concentration gradient and draining back into the venous system at the level of the superior sagittal durovenous sinus. So now, what goes wrong and what kind of CSF flow and related abnormalities are pertinent? Well, certainly when there is a disruption of flow, an excess volume, excess CSF pressure, basically one of several forms of hydrocephalus may result, many of them producing dilated ventricles that are increasing their size in response to either the excess volume or excess CSF pressure. So now, there fundamentally are 2 forms of hydrocephalus. The first one is illustrated on this slide, the next one on the next one. A communicating hydrocephalus is generally caused by either over-secretion of cerebrospinal fluid or a lack of absorption of cerebrospinal fluid from the subarachnoid space. It's generally not caused by an obstruction of CSF flow inside the ventricles. So now, one cause of over-secretion might be a choroid plexus tumor, a papilloma. A disruption of CSF flow in the subarachnoid space by a tumor might limit its flow. Forms of meningitis may limit absorption of CSF back into the superior sagittal sinus. And generally, that's the most common cause of a communicating hydrocephalus, a limiting of CSF absorption back into the venous system. So now, in contrast, on the next figure, a non-communicating hydrocephalus is specifically caused by an obstruction to CSF flow at some point inside the ventricular system or at an opening or channel that interconnects the ventricles inside the central nervous system. So now, common sites where a non-communicating hydrocephalus will develop will be at one or more of the foramina of Monro, limiting flow from a lateral ventricle into a third ventricle. In that case, the enlarged ventricle will only be one or the other of the lateral ventricles. Probably the most common site of a non-communicating hydrocephalus is caused by obstruction of the cerebral aqueduct in the midbrain. In this case, all 3, both of the lateral ventricles and the third ventricle, will become enlarged. And finally, the other cause of a non-communicating hydrocephalus obstruction will be at one or more of the openings that exit from the fourth ventricle. So in essence, in all of these cases, CSF is prevented from exiting through the foramina in the fourth ventricle and limiting CSF flow or drainage into the subarachnoid space. Finally, kind of a subset of a communicating hydrocephalus is known as normal pressure hydrocephalus. And this basically is classically caused when CSF is not absorbed properly at the level of the arachnoid granulations. Their grossly visible structures are known as villi, and the ventricles become enlarged, pressing the available cortex, particularly inside the lateral ventricles and the fourth ventricle, against the skull. This is the most common cause of hydrocephalus that's seen in elderly patients. And typically the elderly patients that present with this will have forms of confusion and a form of dementia in a way. They classically have what is known as an apraxic gait. It's also known as a magnetic gait, where these patients seem to walk as if their feet are stuck to the floor. And classically, they also present with urinary incontinence as well. So let's move on now that we've talked about CSF abnormalities, and just talk generally about the autonomic nervous system. This has obviously been reviewed elsewhere in the gross anatomy section, but we need to make a couple important points relative to how the central nervous system controls autonomic function. First, by way of review, we certainly know that the function of the autonomic nervous system is to provide a motor innervation of three kinds of histologic tissue: all of the smooth muscle in the body, modified cardiac muscle, the conduction system of the heart, as well as cardiac muscle, and all of the glands of the body. And it's certainly well known that the autonomic nervous system consists of two separate, parallel, somewhat antagonistic subdivisions: the sympathetic division and the parasympathetic division. So now the fundamentals of this are these, is that all autonomic pathways, regardless of whether they are sympathetic or parasympathetic, utilize two neurons. And the points are made at the bottom of the slide. The preganglionic autonomic neuron is a neural tube cell because its neuron cell body is found within the confines of the central nervous system. The postganglionic neuron, since it has its cell body in a ganglion in the peripheral nervous system, is derived from neural crests. So looking at it embryologically, we can see that all autonomic pathways are a combination of a neural tube cell linked synaptically with a neural crest cell. So now, let's look at the sympathetic outflow, locations of its preganglionic neurons, and the sites where its preganglionic axons synapse. First off, very important point with regard to central nervous system function is all of the preganglionic sympathetic cell bodies, as indicated on this diagram, are found in spinal cord segments continuously from the T1 spinal cord segment to the L2 spinal cord segment. You could see them illustrated here. So basically, the spinal cord, certain segments of the spinal cord contain all of the preganglionic sympathetic neurons. The motor axons of these sympathetic neurons leave the spinal cord in ventral roots of spinal nerves from T1 to L2, and their goal in life is to synapse in one of two different kinds of sympathetic ganglia. They synapse either in the paravertebral sympathetic chain of ganglia, which you can see extending parallel and immediately adjacent to the spinal cord, or preganglionic sympathetic axons may synapse in prevertebral ganglia, which are only found below the diaphragm and basically are involved in the sympathetic innervation of abdominal and pelvic viscera. So now the important point to be made on here is the fact that, or how the sympathetic innervation reaches structures in the face, the scalp, and the orbit. This is a very important pathway not only for peripheral neuropathies, but as we'll see, for central nervous system conditions as well. So again, recognize that the face, scalp, and orbit are innervated by a sympathetic pathway that begins with a preganglionic neuron at T1. Its axon leaves in the T1 ventral root, and basically it enters the sympathetic trunk at the level of the T1 cervicothoracic or stellate ganglion, ascends the sympathetic trunk, as you can see here, synapses in the superior cervical ganglion at the top of the trunk, and then the postganglionic sympathetic axons are distributed to face, scalp, and orbit. So in these regions in the face, they're certainly innervating sweat glands in the face and scalp, and this autonomic pathway is providing the innervation of two smooth muscles that are in the orbit, the muscle which dilates the pupil and the superior tarsal muscle, which helps you elevate your eyelid in a sympathetic response. So now the important point about this pathway, as well as all other preganglionic sympathetic neurons, is they are driven by descending hypothalamic fibers that are illustrated in red, whose cell bodies are found in the hypothalamus. And since that's the case, the axons must leave the hypothalamus, course all the way through the length of the brainstem, down through the cervical part of the spinal cord, to synapse with and drive all of the preganglionic sympathetic neurons from T1 to L2. So now, we point this out to make the illustration that, as indicated here, any lesion that occurs at one of those black arrows that disrupts the sympathetic pathway that participates in the sympathetic innervation of face, scalp, and orbit will result in Horner syndrome. Certainly, there are lots of causes of Horner syndrome that result with a lesion to sympathetic pathways in the peripheral nervous system. For example, lesioning the T1 spinal nerve, or lesions that affect parts of the cervical paravertebral trunk at to or near the superior cervical ganglion. The point that we want to make here, in addition, is that patients can develop a central Horner syndrome if those descending hypothalamic fibers are lesioned, particularly in the brainstem and the upper part of the spinal cord. So now, localizing factor with regard to Horner syndrome that's going to be valuable to us is that, note as we indicated at the bottom of this figure, that Horner syndrome signs and symptoms, which include meiosis, ptosis, and anhidrosis, those signs and symptoms are always going to be seen ipsilateral to the side of a lesion. The lesion can certainly be lots of places, either inside or outside the central nervous system, but the signs and symptoms of Horner syndrome are always going to be ipsilateral to the side of the lesion. So now, let's take this one step further and just illustrate some of the features of the parasympathetic component of the autonomic nervous system. Much of this we'll deal with elsewhere when we review the cranial nerves, because the point to be made on here is that parasympathetic preganglionic neurons, instead of having an exclusive thoracolumbar location as they did for sympathetics, have what we refer to as a craniosacral distribution, meaning that many of the preganglionic parasympathetic neurons are found in different parts of the brainstem and exit with cranial nerves 3, 7, 9, or 10. The other location of preganglionic parasympathetic neurons, as indicated here, are found at sacral spinal cord segments S2, S3, and S4. And as they exit those spinal cord segments, they basically are known as pelvic splanchnic nerves. So let's summarize the connectional features of the parasympathetic nervous system, and this is illustrated on the next slide. Fundamentally, the cranial nerves 3, 7, and 9 basically contain preganglionic parasympathetic axons that are going to synapse in 4 named ganglia that are situated in various parts of the head. Their job overall, and we'll see this in more detail when we deal with cranial nerves, is to innervate the salivary glands, submandibular, sublingual, and parotid, all of the mucous glands of the oral and nasal cavity, and again, 2 smooth muscles in the orbit, the sphincter pupillae, which constricts the pupil in response to light, and the ciliary muscle. Cranial nerve 10, as well as the pelvic splanchnic nerves, exit from their cell bodies and synapse in terminal ganglia. Terminal ganglia are typically found in or near the wall of a visceral structure being innervated. The postganglionic axons from those terminal ganglia that are associated with vagus nerve function provide the parasympathetic innervation to the heart, lungs, and foregut and midgut glands and smooth muscle. Pelvic splanchnic nerves also synapse in terminal ganglia. They're going to provide the parasympathetic innervation to hindgut glands and smooth muscle, as well as the smooth muscle of pelvic viscera, largely including the bladder, and provide the innervation to erectile tissue. Note that it is the terminal ganglia that are linked together with pelvic splanchnic nerves, most commonly in the wall of the rectum, that are missing in infants born with aganglionic megacolon or Hirschsprung's disease. So up to this point, basically, our summary has been directed towards, so far, neurulation, the development of the neural tube, the significance of the neural tube defects, and your ability to distinguish between a fetus that has anencephaly versus different forms of spina bifida. Then we also addressed CSF flow, the implications of communicating versus non-communicating and normal pressure hydrocephalus. We talked about the functional aspects of neurons and the implications for transport processes, anterograde and retrograde axonal transport, as well as the differences in the capacity of an axon to regenerate, depending on whether it's myelinated by oligodendrocytes or Schwann cells. And then finally, we documented the differences in the components of the autonomic nervous system, leaving much of its detail for discussion of the nerves themselves, particularly cranial nerves, and pointed out the significance of Hirschsprung's disease for terminal ganglia associated with pelvic splanchnic nerves, as well as the many different sites where a lesion can result in central or peripheral Horner syndrome.